Hi, I'm Ushma Neal with the Journal of Clinical Investigation, and I'd like to welcome you to the next in our series of conversations with giants in medicine. Today we're speaking with Paul Marks. Marks is recognized as a leader in the cancer world, as a scientist, clinician, and a leader, as he served as president and CEO of Memorial Sloan-Kettering between 1980 and 1999. He's also led the discovery, testing, and recent approval of SAHA, the treatment for cutaneous T-cell lymphoma. SAHA and other histone deacetylase inhibitors are now undergoing trials for a broad array of cancers. We look forward to hearing about your path from the neighborhoods in Brooklyn to training with Nobel laureates and the view from behind the desk at the most powerful desk at Memorial Sloan-Kettering.
Right.
So can you tell us a little bit about your path to medical school and the influence of your parents and your grandparents?
Yeah. So one of the ironies in my life is that I was raised in a small coal town in Schuylkill County, Ashland, Pennsylvania, hard coal region. And my mother died when I was about 5 and a half. And the irony there is that that brought me to New York. I think otherwise I would have been like all my cousins in that area, remained. It's really quite remarkable that with probably one exception, all my cousins have come back to or stayed in Schuylkill County in the small towns there. So when I came to live with my father's parents in Brooklyn, it was a very crowded household and I guess not too comfortable. We had my grandparents and my father's older sister and younger brother all living in a sort of two-bedroom apartment. I think the really very significant event in my life was a teacher I had in high school, Conrad Sapphire, who sort of, I think it's fair to say, adopted me in my junior year at high school. He had lost his only son, who was a Cornell Medical School graduate, in Guadalcanal. And he was sort of determined to try to get me to go to medical school. And of course, this was still in high school. But he was very instrumental in helping me focus on how to even apply to college. I would say that my grandparents weren't exactly interested one way or the other, and my father wasn't around at the time. So he suggested I apply to several schools. The issue, frankly, was that Columbia was the only place in which I could afford to get to, a 5-cent subway ride. And I was admitted to Columbia College, which was a really cultural and life-changing experience. And it was a very exciting time, and we were at war in early '43. And then the so-called Naval Officer Training Program came on the campus, and I applied for and was accepted into that program. And that, by '45, I really completed the requirements for the degree, and the Navy had a program of wanting to send candidates to medical school because they were concerned about having enough physicians in the service. And I took the exam for that and got assigned to PNS. So I was really on a path that was relatively without trauma in applying both to college and medical school. And I entered medical school under the Navy auspices, but by October of '45, the war was over, and we became civilians, and then I got the GI Bill of Rights, which financed most of the rest of my medical education.
Now, during medical school, did you ever see a scientific bent towards your studies, or were you focused on being a clinician?
Actually, my interest in research started in college. My roommate in college was Josh Lederberg, a future Nobel laureate. And Josh was working with Francis Ryan, who was a very prominent geneticist, professor, and he really literally dragged me into the lab. So while I was an undergraduate at college, I started working on a problem in Ryan's lab and really got hooked. I really loved the idea of trying to answer a question to which we didn't immediately have an obvious answer. So that was my introduction to research. And then when I got to medical school, I pretty much took every advantage, electives, etc., to do a research electives with various professors. My first one was with DeWitt Stetton, who was professor of biochemistry. That was mainly a literature search. But the first hands-on lab elective, I think I started in my sophomore year and continued. My junior was with Erwin Chargaff, famously the one who discovered the nucleotides in DNA, frustrated that he wasn't recognized with the prize when Watson and Crick got it. But Chargaff at the time was working on trying to discover the genetically determined enzyme defect in Gaucher's disease. So that was an interesting experience, really very intense. He was a very tough mentor, but good. Of course, we never really discovered what we were looking for, but it was a good experience. And I pretty much realized that I was more interested in research than I was in clinical medicine.
Now, you also did 2 postdoctoral fellowships,
Right.
perhaps not one after the other, one with Arthur Kornberg at the NIH and then one with Jacques Monod at the Pasteur
Right.
Institute. How did those 2 experiences shape your...
Well, on graduation, I did do a 2-year, at that time, 2-year internship. But it was interrupted in the second year because of the Korean War, and they started recruiting. And I was extremely fortunate because at that time they were also recruiting for the new National Institute's Clinical Center Research Program. And I got recommended by the then professor of medicine, Robert Lerb, to James Shannon, who was head of the NIH in the early 50s. And I reported, I think it was probably February of, 52? Yeah, I think it was February 52, to the NIH, presumably to staff the Clinical Research Center. But the Clinical Research Center was still being constructed. So I had to find something to do, and it was suggested that maybe I should walk into Kornberg's lab. He was, I'll never forget it, in Building 3, which was in front of the main administrative building at NIH. And the anecdote there that I thought that I'll never forget was somehow or other Arthur interviewed me. He took me into the conference room and went to the blackboard and started putting stuff on the blackboard, and he said, okay, this is what you're going to work on. I didn't have a clue what he was talking about. And he said, I also want you to know that you're the first MD that I've ever had as a postdoc. And that was it. So I decided I better go to the library. But first I looked up Kornberg. Of course, Kornberg himself was an MD. He had graduated from the University of Rochester. And I had a great time, but by the summer, Kornberg was off to being, I think, professor of microbiology at the Washington University in St. Louis. So Werner Hoeker took over as the head of the lab, and he was also a terrific mentor.
And then how did you land in Jacques Monod's lab? Okay,
so when I finished at the NIH in '55 and went back to Columbia, we had been working on what was then called an alternate or new pathway of glucose metabolism, the so-called pentose phosphate pathway. And we had been working with spinach, looking at, isolating enzymes and isolating substrates and so on. So I thought it would be interesting to see whether that pathway existed in human mammalian cells. And the easiest cells for me to look at would be red cells because they were very easily available. And I did, in fact, demonstrate that this pentose phosphate pathway operated in red blood cells. But one of the sort of unexpected findings was that the first enzyme in that pathway is called glucose 6-phosphate dehydrogenase. And we had developed a assay for the enzyme, and I started looking at different individuals. We would draw blood on each other, and I drew blood on my technician. My laboratory was a force of two, my technician and myself at the time. And she turned out to have a low level of G6PD. At first I thought, God, she doesn't know how to do the assay. So I did it, and I still got a low level. And also at the time, I was collaborating with somebody at Stanford, Toby Gross, and she was interested in a hemolytic anemia that she saw occasionally in a large Greek family. And it turned out that they also had G6PD deficiency. And as we studied the several generations, it was clear that we had, on one hand, a genetically determined deficiency, and on the second, we had enough individuals in the 3 generations that we were able to study to strongly suggest it was sex-linked. And at the time, I had the studies that showed that G6PD was a genetically determined deficiency, and the issue was, how does a genetic defect, a mutation in the DNA, cause a abnormal protein? And it was clear that great things were happening, but not at Columbia, in the world of molecular biology. And one of the hottest labs at the time was in the Pasteur with Monod and Jacob and Leboeuf. And I applied for a visiting scientist position, it was called. It was sort of beyond the postdoc phase, since I was a faculty member, and was accepted, got a Commonwealth Fund fellowship, and off we went to Paris. And we had a great experience. And Monod turned out to be a great mentor. He was tough, and if you thought you were good, it was great. If you thought you weren't, you were miserable. Unfortunately, he thought I was okay.
So in looking at your CV of over 400 scientific articles, in addition to your general articles about policy, I couldn't see necessarily a common thread. You seem to have studied everything from protein synthesis and globin biology to adipose tissue to then more recently in the latter half of your career, more cancer biology.
Right.
How is it that you were inspired by so many different scientific topics?
So the work on lipid metabolism and so on, I had done soon after I came back to Columbia when I was doing the work on G6PD, it was an interest of the head of our cancer group, Al Gellhorn, and I really worked with him on it. I was sort of the main person doing the lab work, and he was interested. But it wasn't a mainstream interest of mine. The mainstream interest was on the genetically determined defect and so on. So when I came back from Paris, I turned to looking at a possible human disease where what we had discovered in Paris, which was the mammalian messenger RNA and its stability and the role it played in transfer of information from DNA to ribosomes and protein synthesis, I thought that thalassemia, which we knew represented a defect in globin production. In fact, there was a controversy at the time. That was very interesting. There were papers published by important people like Carl Moore, who was leading professor of hematology at Washington University, that thought that thalassemia was fundamentally a defect in iron metabolism. And then there was some evidence that it probably was a defect in globin synthesis, but it wasn't very strong. So I looked at the synthesis of globin. We developed, following on our work in Paris, cell-free system to examine globin synthesis using ribosomes with their RNA from patients and from normal. And what we quickly found was that if the ribosomes of the thalassemia patients were fine, it was their messenger RNA that was defective. And so it was clear that the defect was in the DNA translated into defective message, which led to actually a decrease in what we were studying, which was the synthesis of beta globin. And those studies on globin synthesis were central in my lab at the time when I read this report from Charlotte Friend, who found that the solvent dimethyl sulfoxide induced her virally transformed mouse cells to produce a red pigment. Actually, Charlotte had called me and asked me, what human proteins are red, and I said there's only one. And it was clear that she had discovered that dimethyl sulfoxide induced these viral transformed cells to produce hemoglobin. So this looked like an interesting model to pursue further the whole regulation of the globin gene expression. And we got the cells from Charlotte, and she was very generous, and we started looking at them. And the one thing that struck me even more interesting at the time was that while DMSO induced globin and hemoglobin synthesis, it also arrested the growth of these cancer cells. It didn't induce their death, it just arrested their growth. And that, I thought, was an extremely interesting phenomenon. And so I decided to pursue the mechanism of DMSO induction of transformed cell growth arrest, and that it took me the next 20 years. And so with Ron Breslow, who was professor of chemistry at Columbia at the time, and a group of very talented postdocs over the next years, including when I went to Sloan-Kettering Memorial, my lab continued to try to understand the mechanism of action of these agents that clearly could not only induce the arrest of a great variety of cells. In fact, our agents, we gave them to the NCI once, and they screened it against their 60-plus transformed cell lines, and it was remarkable that these agents, which eventually the lead compound was SAHA, inhibited to a greater or lesser extent almost every transformed cell line against which we tested it. And but the issue of the mechanism of action of these agents still remained a challenge until the early 90s, when a Japanese group reported that TSA, which is a natural fungicidal compound, could arrest the growth of the same cells, these viral transformed cells we were working with. And that it led to the accumulation of acetylated histones. They had not yet identified the target, which were the histone deacetylase enzymes themselves, but reading that led us, to be able to reproduce their results with our compound, which was SAHA and related. And we pursued the mechanism. One of my postdocs was very involved in this, Victoria Rochon, and we're able to identify the targets as these HDAC enzymes. And over the years, of course, several things happened. Number one, we were able to really gain a lot of insight into the mechanism of action. There are 11 different zinc-dependent HDACs in humans, and SAHA inhibits 4 of them, so-called class 1, HDAC 1, 2, 3, and 6. And it also turned out that, and this was, in my view, personally, very exciting, because at this point, we thought, okay, let's see if it works in animals. Let's see if it works in vivo. We knew it worked in vitro. So we took a human prostate cancer transplanted animal mouse, and we gave it SAHA. And not only didn't we kill the animals, but we stopped the growth of the tumor. And we stopped the growth of 100% in the mice, which of course is a far cry from what we ever saw in human subjects. But it was really a very potent, striking result. And so this was in the late 90s. And at that point, I was, at Memorial was a place where it was, there was a whole group focused on bringing anti-cancer drugs to clinical tests. So with their help, we did the necessary preclinical studies to get FDA approval to go into the clinic. And I think it's one of the rare examples of an anti-cancer drug that was developed entirely up to including actually phase 2 clinical trials in an academic setting. And then I guess it was so we started these clinical trials in 2001. At one point, it was clear that they were going to be expensive, and the board decided that they didn't want to go for it. So we started this company, Aton, and raised venture capital money, which permitted us to go into phase 2 clinical trials. And they were carried out mainly by Madeline Duvik as the leader at MD Anderson. Mm-hmm. And the clinical trials were sufficiently impressive so that the FDA put us into registration based on these phase 2 clinical trials primarily targeted against patients with cutaneous T-cell lymphoma, which was, it's a rare cancer, but it was at the time one that was extremely difficult to treat. And patients who had it had a miserable life. It was at this point that the little company attracted a lot of pharmaceutical company interest. And, after a period of negotiation in which the venture capitalists really played the major role, and they were very anxious to get out ahead of the game. And so Merck bought the property, bought the license from Columbia and Sloan-Kettering.
Now I want to take you back a little bit to some of the early days of your career, because at the peak of your most exciting research findings between 67 and 71, you served as editor-in-chief of the JCI.
Right.
Can you talk a little bit about how that experience affected you?
Well, in this, so in the 60s, late 60s and so on, among other things, I was very involved in the American Society of Clinical Investigation, had a lot of buddies and looked forward to the annual meeting and so on. And the editorship of the journal came up, and I was approached by members of the search committee to see if I would be interested. And I think I hesitated, obviously, for a little while, but I looked upon it as an attractive opportunity to really get a broader perspective on what was going on in the research world. My tenure as editor, I think, lasted about 4 years, and it really was, I think, a very rewarding experience personally. I had a great group of associate editors, and we used to meet religiously every Wednesday afternoon. We'd get together around 2 o'clock in the afternoon and go over all the potential manuscripts that we would consider for acceptance or possible acceptance of the journal. But it was really a journal club, which spanned very broad areas of both basic and clinical research, and it was a terrific learning experience.
Shortly after that, or concurrent with the end of your editorship, you were tapped to become the dean of Columbia's College of Physicians and Surgeons, but you famously submitted your letter of resignation the day you started to count 3 years later. So why did you approach it that way?
Well, because first of all, I never thought I really wanted to get into administration. Strange, considering what happened. When I was actually chairman of the search committee to find a successor to Merritt, who had resigned as dean in the general convulsions, if you will, that occurred at Columbia in the post-68 student student uprisings, if you want to call it, occupation of the university, etc. And Columbia had been a top-down administration, and the events of the 68 and 69 completely changed the university. So we had this search committee, and we came up with 3 candidates, 3 outstanding candidates, we thought. And we had this very complicated relationship between the medical school and Presbyterian Hospital. And the head of Presbyterian Hospital was a guy named Augustus Long, who was chairman of Texaco, and he was chairman of the hospital board. And the bottom line was that he found all of our 3 candidates unacceptable. And although there is no contractual obligation on the part of the medical school to have the hospital or the chairman of the board of the hospital consent to who would be dean, at the time, the leadership of the university, I think, was, in retrospect, not ready to stand up to him. So all our 3 nominees were knocked down by him. And this was frustrating because we had worked for over a year on this. And without my knowledge, members of the search committee went to the then president of the university, or acting president, Cordier, and suggested he invite me to be dean. I really was ambivalent about it at the time, but felt that the school was really needing leadership. We didn't have a dean. 13 of the departments, including all the major clinical departments and some of the preclinical, were without chairman, and this was my school. So I agreed to do it, but I thought that I could sort of take a somewhat partial leave of absence from my research for 3 years, but beyond that I really would lose it. And so when I accepted the position, I also sent my letter of resignation to McGill. And I spent the next 3 years focused as dean largely on recruitment, which was very successful in retrospect, including 2 future Nobel laureates, Eric Kandel and Richard Axel. And we were able to recruit really outstanding people for the chair of the department, and I think the school took on a different trajectory.
So then after that period, can you tell me why you decided to instead take on the presidency at MSKCC?
So I was really very interested in the cancer problem. I had, by that time, served on the President's Cancer Panel with Schmidt, gotten involved sort of both at the national level and particularly at the school in developing our cancer programs. And frankly, it was a very uphill struggle to develop what I would consider a cutting-edge cancer program, particularly a clinical cancer program at Columbia Presbyterian at the time. Fundamentally, the clinical departments I don't want to say weren't interested, but they didn't exhibit any interest. For example, medicine and surgery had no oncologists, real oncologists, identified as such in the department, and certainly not at a senior level. And it was very frustrating to try and develop what I would consider a cutting-edge cancer center. We had very strong research in certain areas, but our clinical programs were very weak. And at this point, I guess it was late '79, I got this call from Lawrence Rockefeller. And I thought that the challenge was extremely interesting because I felt I could both work at an institutional level as well as the laboratory bench in areas of cancer research and cancer care. And I think in retrospect it turned out to be a terrific opportunity.
Now, when you arrived there, you set about making a lot of changes. How were they received?
Well, at the level of the board, extremely well, and that was critical. I mean, both Lawrence Rockefeller, who was chairman, and Benno Schmidt, vice chairman of the board, and the board members were extremely supportive. It was absolutely, in retrospect, indispensable. I didn't realize how important, their support was to getting things done that we wanted to. They were not risk averse. What their standard was the best care we could ever provide and the best research. And both, but Benno in particular, had this incredible network. And, I would come up with a proposal to do X, Y, Z or recruit, and he'd say, oh, and I'd tell him about it because usually it required something that the board had to approve, and he would call me back almost routinely a day or two later, and he said he checked with Kornberg or he checked with Paul Berg or, one of the many people he had contact. He said, terrific, go. So that was very, very important. And I think that the most important thing we accomplished was the recruitment of really outstanding people, both in the clinical and science areas. And then programmatically, they were gung-ho for doing things better and different. And we had strong support from the board and now from the clinical faculty and the science programs, to develop programs like the breast center, which was unique at the time. Now, of course, every cancer center has a breast center. But to focus on women with breast cancer who are walking well and put them in a facility where it was tailored to their needs was innovative. And the pain program was a unique program focused on pain control. You would think it's obvious in retrospect, but big issue in treating cancer patients. Jimmy Holland's program in the psycho-oncology was the first in the country. We developed the so-called disease management teams in the early 80s, and that was, I don't know if we were the first, but we were among the earliest to really cut across disciplines. In other words, we put together a team focused on a cancer that the patient had. So you'd have the surgeon, the medical oncologist, the radiologist, the radiotherapist, et cetera, whose expertise, and that was, of course, I think one of the unusual, if not unique things still about Memorial is the depth and breadth of the expertise, as you will learn. So that we were able to put together these disease management teams that focused on prostate cancer, breast, colon, lung, sarcoma, and so on. And this model, evolved and worked extremely well. And we just, as opportunities or new directions indicated, we just grew. We injected genetic counseling. Of course, my wife was very influential in that, but that became a major part. And that was sort of curious too, because when she first got into the programs at Sarah Lawrence and training genetic counselors, cancer was not a big issue. But by the late 80s and early 90s, cancer was probably the biggest issue in terms of genetically at risk individuals, and the whole understanding of the role of genetic alterations in cancer. So it was a very exciting 19 to almost 20 years. And I again think that the two really things was the recruitment of these outstanding people with whom it was really exciting to work, and the programs. And the programs really depended on the trustees' support, which was always there. And that was in striking contrast, again, to Columbia Presbyterian. The trustees of the university are very remote from the medical school. Mm-hmm. And frankly, I had a conversation yesterday with a member of the Columbia board, and nothing much has changed. The first meeting the Columbia University trustees ever had on the medical school campus was during my tenure as dean, in the 200-year history of the school.
Now, you mentioned your wife, Joan Marks, but I also want to bring up your son, Andrew Marks, who has followed in some of your footsteps as becoming a medical doctor and pursuing that training, though in cardiology, and also filling a professorship at Columbia University. He also acted as editor-in-chief of the JCI. But he has told me that you have dissuaded him from taking on an administrative or leadership role in other institutions. Why? Well,
I'm prejudiced, but I think he's an extremely talented scientist and has been extremely productive. And there's no question that I feel, and I think it's real, that I made compromises in my research. I don't know what I could have accomplished that I didn't, but I sort of feel that I could have accomplished a lot more if I were, as I have been for the last 12 years since I focused on the research. I mean, the fact is that bringing SAHA out to the clinic, et cetera, was something that I was able to do after I stepped down as president. And I feel that he has terrific research accomplishments right now and is on to a very exciting area, which is a little unusual. I think his contributions have been very, very important. And I think for him to take on a major administrative role right now, which he has been tempted to do from time to time, would perforce compromise his focus on research. So anyway, all I can do is advise him. I mean, I'm not going to be able to prevent him from doing what he wants to do.
He's also spoken to me about what an excellent mentor you have been to him, and you must have trained hundreds of people during your scientific career as you continue to do. So what do you think are some of the critical components of being a good mentor? Well,
I think what I learned from my mentors was setting the right example. I think, rigorously viewing the question you ask. I mean, successful research begins with asking an important question that is soluble. I think Medawar was the one who wrote, the Nobel laureate in immunology, wrote a little book which was very influential in my career called The Art of the Soluble. And that focuses on the fact that, good research really begins with asking the question in a precise fashion which is soluble. And so I think that's one of the most important things that a mentor can try to instill in students and postdocs and so on. And I think that really rigorously looking at the data, primary data. I mean, my habit to this day is to really sit down with my students and postdocs in the lab and really pore over their gels, their data with regard to sort of all aspects of the thing. And I think there's no substitute for making them understand that, where the answers lie are in your data and the way you fashion your experiments to produce the data. And the more successful your research, the more it will reflect the fact that you really are on target in the way you've asked the question and tried to answer it. So I think that's what you try to instill in your students, is an approach to research. And of course, underlying it all, and I've been extremely fortunate in this, I mean, you expect when you're doing research to have negative results. You expect to have periods when just nothing is working the way you thought it might. But that shouldn't discourage you from, marching on. And the last thing you want to do is compromise on the way you try to get those data. I mean, it's got to be rigorous, it's got to be reproducible, which is the key, and you're going to have to expect that a fair amount of the time you're not going to succeed. But you can't be discouraged. You hope that the majority of the time you'll be getting the right answers, or good answers.
So if you were to do something other than science and medicine, what do you think it would have been or would be?
I've thought about that a lot. Now, I guess my second choice would be to be a curator in a good museum. We're very interested in art, and as you know, my daughter and my younger son are both involved in the art world very heavily, and we've always been interested in that. And from the time when we were young, didn't have babysitters and so on, we dragged them through every museum in every country we visited. So art has always been a passion, and still is. And I would say that the two ways I find relaxing, and I must say that from time to time, I used to cop out from my office at Memorial and run to the Met, are to go to a museum and just walk through, or to play tennis. And since my back gave way, the museums have taken over.
Thanks for joining us today.
Thank you.